Good afternoon. We're going to start the session. I've been waiting a couple of minutes, but this is time to start. First of all, I want to thank the Institute of Neuroscience, especially the organizing committee, for allowing us, the scientific committee, to invite such reference scientists yesterday and today. I want to remember the people that is attending online that they will be able also to make questions. We will be reading them. And now it is my pleasure to introduce you to Alessandro Gozzi. He's a senior scientist and director of the Functional Neuroimaging Laboratory at the Italian Institute of Technology in Rovereto, Italy. He holds a ERC starting grant about the neural basis of functional disconnectivity in brain disorders. And I can't wait to hear you.
Thank you so much. And yeah, thanks for being here. Thanks for having me here. So, okay, so let me just keep track of time. Yeah, so today I'm going to talk about this, what you read there, so biological decoding of brain functional disconnectivity. And that's, I'm fully aware that given that there's such an heterogeneous audience, what's written up there might not mean much to you. And so in the first 5 minutes or so of my talk, I've taken the liberty to illustrate in detail, just to make sure that everyone is on board, to illustrate what I mean with this title. I'm going to parse it to you. So if you understand the research question, hopefully you will be able to follow what I'll be showing. I promise it's simple stuff. Everyone can really understand this. And hopefully it's going to be perhaps of interest to such a varied audience. All right, so at the cost of appearing condescending, I'm going to explain what this is, but you know all too well. So my talk is going to talk about functional connectivity. And functional connectivity is a very deceiving concept. And that's why, as I said, I'm going to invest 5 minutes to explain what that means in the context, in the realm of system neuroscience. And so in order to understand this well, and once again, apologies for those of you who know already, but that's important. In order to understand this well, we need to understand how functional connectivity is measured in the brain. And to do that, we use fMRI. You've seen many talks this morning. fMRI is this thing here, right? And it's like a canonical acception. So there's like non-invasive way of measuring brain activity. In this case here, the subject is into the scanner. It's presented with like a visual stimulus, okay? And, quite... how to say, according to expectation of what we know, visual stimulus activates, the visual cortex, which is something that everyone knows, everyone agrees with. And so this way, fMRI is being used widely, right, to, for example, locate functions, right, to see what kind of stimuli activates what kind of region. However, if we want to use fMRI to measure functional connectivity, we have to use a different flavor of MRIs. And in order to do that, we have to use a couple of simple tricks, very simple. The first one is this one here. We have to get rid of this evoked activity or task that administration. So what we do, very simply, we ask our people inside the scanner to stay at rest, don't move, try to keep awake. That's difficult, but we have to, or at least very important. And so as a result of that, or this very simple trick, what you get is this sort of like uneventful, perhaps boring signal, right? Because nothing is happening, there's no stimulation. But look what happens if you just extend this recording to 1 minute or 2 minutes. You see what's happening? You have this slow symphony of activity that is emerging out of this resting signal. And these slow fluctuations lie at the very basis of what functional connectivity is. So this is like a real-world recording in the brain of a subject of this sort of very slow activity, the one I just showed you. And by looking at this, you can really understand what functional connectivity with fMRI is. It is basically an index of this coordination that we see in this signal. As you can see, if you just look these ups and downs of signals, they are strongly coordinated in space. Look how symmetric they are, and also in time, right? They rise and fall. And so functional connectivity, this is really the take-home message for you. And once again, apologies for those who knew it, right? This is really straightforward, but functional connectivity, in spite of its name, is a measure of synchronicity. It's not a measure of connectivity, okay? Then we can discuss why it's called like that. That's not important. So keep that in mind. And this synchronicity can be measured in many ways. The most simple one, perhaps it's for sure imperfect, but that's what people use, and we use it as well. It's just a simple correlation between these time series across time. If they are correlated, they go up and down together, you can put like a yellow voxel, and you can then get this sort of like symphony maps, right? Like when, you know, voxels that have the same colors, they're kind of playing the same tune, right? And you can change colors and see whether there are different tunes in the brain, so to speak. Okay, so that's it for my introduction. So this is just a long introduction to tell you that fMRI connectivity is a measure of synchronicity. That's very important to understand what I'm going to show you. And clearly now, I'm not going to review the whole field, right? But I'm just going to tell you that this very simple approach is very powerful. It has really, increased our understanding of the brain works as an integrated network. And these signals are at the basis of who we are as individuals, because these symphony signatures are really different across people, but they're also at the basis of some cognitive functions, and also they change, with development as we age and so on and so forth. And so it might not be surprising to you at this stage to learn, and probably something that you also see in this workshop, that functional connectivity is almost invariably disrupted in every brain disorders. If you have your favorite one, Google it, PubMed it, you will see functional connectivity is altered, anxiety, depression, schizophrenia, Parkinson, you name it. And here, this is really the basis of the research I'm going to show you today, because, okay, there's so much research, but this is so far, up to date, pretty much phenomenological research, which means that we have these huge databases with these scans, but we have failed to tap into the information that is there. So it's phenomenological, meaning that, there's a description in depression, you have lower connectivity, which is synchrony between some regions, but why? What does it mean? What are the molecular causes for that? And so, we are left with these big open questions, and the research I'm going to show you today, which is so far unpublished, so I'm very happy also to hear what you think. Try to tackle this question. Can we just use this information, these maps, to learn, to infer something about the underlying pathology, okay? And I'm not claiming we managed to do that, but, perhaps we made a little progress, and as I said, I'm very happy to share it with you and to see what you think. Okay, this was my introduction. I hope that it worked out. Okay, outline of my talk. There are two parts, short parts. One is about how we tried to molecularly decode what I define fMRI disconnectivity. And there's a terminology alert here. Disconnectivity here stands for dysfunctional connectivity, or functional connectivity. Sorry for the repetition, but this means that it could be like higher connectivity, so higher synchrony, or lower synchrony, okay? But I call them both disconnectivity to simplify. And there's also here hidden away in autism. And if you read here, it's because autism is just an example. You don't need to be, interested in autism. You don't need to know anything about autism. You can, if you're not into autism research, which I'm sure you're not, that's the case, just focus on the approach. This you can apply once again if you have a favorite disease. If there's such thing as a favorite disease, you can apply this to that. In the second part, and the final part, I'm going to instead show what we did in the context of trying to understand the physiology of these alterations. What do they mean in terms of, brain rhythms, for example, okay? And that's what I'm going to talk about. As I told you, this is going to be about autism as an example, okay? And all you need to know about autism, I'm going to tell you now, and I'm sure you know. Autism is a developmental disorder, right? So early development, and it is diagnosed behaviorally, okay? There are these core behaviors, restricted and repetitive behaviors, socio-communicative, communication alterations. And it is also, it's beginning to be quite prevalent now. Latest stats suggest that it's one child out of 100, depending on the country, is affected. That's all you need to know, okay? But why am I showing you this? Well, I'm showing you this because when fMRI connectivity became to be prominent, it is so easy to do, right? The inconfessable dream of every brain imager was, okay, maybe, given that autism is diagnosed behaviorally, maybe if I do this sort of scanning, which is very simple, 10 minutes, no task, nothing, maybe I can see autism in the brain, which means either I can diagnose it, right? Maybe we find something that is specific, or I can even better, I can find like a circuit or a network that is really specific to this sort of like dysfunction. And so, to cut a very long story short, there have been many studies in this field, and the results we got are very interesting. There are examples where there's decreased connectivity, so these regions are out of sync. But there are also studies that show that there is increased connectivity. So these regions, right, they are overly synchronous, like the brain is playing the same tune over and over. And it is also interesting that there are studies showing that there is decreased and increased. And so I'm asking you, can we see autism in the brain? No, we cannot, right? Not yet, for sure. If there is such signature that is specific to autism, we have yet to find it, okay? And this is where the research in my lab started. And what I found really fascinating here, and also shocking, I remember, when I was reading this, because this was a part of me writing grants and, catching up with the literature. And when I was reading these papers, up to, these are old, but, really, even now. And you read the discussion of these papers, basically, what is emerging is that, this is really like something dominant in the field. What is emerging is basically this sort of tune, like people say, okay, we haven't found it in this specific study, like this signature of autism, but we have not found it because fMRI is noisy, there's the reproducibility crisis, and perhaps, there must be such thing, but we should use like better methods. fMRI is not suited to this, which is fine. fMRI is always like criticized, and we know all of that. But it is, as I said, fine, but there's also an alternative, right, hypothesis, which is equally plausible. And the hypothesis that we wanted to test it here is that we have yet to find this signature because there is no such thing. Maybe there is no such signature of autism. Maybe there are different kinds. And the problem is now, how do you tell these two hypotheses apart, right? How can you test this second hypothesis? Well, this second hypothesis, the idea that there is no such signature of disruption that is specific to autism or schizophrenia or depression, is in fact, as a neurobiologist, is supported by what we know about the autism. Autism is a spectrum. This is something you know. I haven't told you, but I'm sure you know. And what's even more important and more relevant to my talk is that autism is amongst the most heritable disorders out there. So the risk to develop autism if you have a child of like, two homozygous twins is like, to have them both basically diagnosed is 90%. So it's really amongst known like complex disorders, one of the most heritable. Well, when you look at the genetics, now there are more than 100 high confidence autism risk mutations identified, and none of them explain more than 1% of the cases. And if you look at them, they are not like, on a single pathway, but they're kind of spread out. So there is so much heterogeneity. So it might very well be, right, that this heterogeneity is kind of impacting brain connectivity, for example, for one phenotype in different ways. But once again, how do you test this, rigorously? Well, what we did in the lab, we leveraged this stuff here. My lab, over the past 15 years, has been, investing a lot in trying to implement these translational measurements across species. And as a matter of fact, if you look at these connectivity networks, to some extent, they are very well conserved also in smaller species, for example, in rodents, okay? This is not to say that rodents are miniature humans, for sure, but, a lot of the organizing principles are really conserved. And if you want to know more about this, check out this recent study where we'll discuss, and we show also the dynamics of these fluctuations is really similar across species. And so we said, you know what? How about using imaging genetics instead of in humans? How about using it in mice? And even though this may seem crazy, it kind of makes sense, and I'm going to explain you why. So the idea here, well, before you get distracted by this, the idea here is simple. We know that autism is strongly genetic. We know that genetics affects connectivity maps. This we know from humans. There are so many studies because our maps are slightly different across, this room. We have subtle differences that are also, driven by our genetics. And so the idea is, okay, maybe we can leverage the genetic heterogeneity of autism as a key for decoding the biology behind it. So the idea is simple. If I just can accrue many mutations in mice, which we can do very easily, we can use every single mutation, as long as these mutations are unrelated, as a tile of a puzzle. And we can see whether we can compose like a landscape or whether those tiles overlap one, onto each other. If they overlap, there's one signature. If they don't, it means there's a spectrum. And why is it interesting and important to do this in the mouse? Well, first of all, because in mice you can control beautifully and tightly environmental variability. Environmental variability affects our networks. Genetic variability affects our networks. In mice, all mice are genetically identical. Environmental variability is controlled, okay? And also in mice, you have a very clear control population. They're called litter mates, the same mice, same, litter, and so on and so forth. Also, you can also control motion, head motion, by the way, which is something that people have been putting into question. And so that's what we did. Over the past 10 years, we've been doing that. We've been acquiring, basically, fMRI connectivity in all these mouse models. These are like recapitulating biological factors that are related to autism, okay? And for each of these, we can map what's the net effect of the mutation or for example, in this case, there are also immune factors. Okay, some results at last. Okay, this is important. You have to pay attention. There's a color coding here throughout my talk. Very simple. Blue means reduce connectivity throughout my talk. And what I'm showing you here is the difference map. This one is the gene that we mutated, which means that these mice that have this mutation have lower connectivity here, and here for all of them. And if you just take a look here, there are some convergences. For example, all these mutations produce blue in these kind of midline regions, which is the default mode network of the mouse. Mice have a default mode network. But if you zoom out, you add more mutations. You see there's a shift between midline to like here more sensory. Some of them don't have anything in the cortex. And then there's the last group, which is my favorite one. And it is my favorite because it's red. And red means more connectivity. Keep that in mind. Why is it my favorite? Because I think you can grasp, right, what's happening here. Here we go from blue to red. That's exactly what I showed you in my first slide before. So this explains very well why in humans, in some studies, we have blue, so less connectivity, red more, or a bit of both. If these were like, autistic people, of course they're not. Mice do not have autism, but if they were like autistic people, when you pick randomly, right, you get a bit of blue, a bit of red, and then you get this inconsistent data in humans. So in my opinion, this explains very well what's happening in humans. And there are a couple of important lessons here. First, etiology, so biology, is a key determinant of this variability we see in humans. Okay, so perhaps we should stop this crazy quest for the single signature. There are many, based on what I'm showing you so far, but there's more to come. But perhaps the most important question here is that now maybe we can really focus on what's converging then. There are possible convergences here, right? And if these convergences, by any chance, were produced by some sort of shared pathological mechanisms, maybe then we have another question. The question is, instead of finding the single signature of autism, we can find maybe different autisms that are, supported by different biological alterations. And so the question here is, why do we have some that are red? Why do we have some that are blue? Are these stochastic changes, or are they an expression of common biology? And if you come to think of it, this is very simple to address, because for these mice, we have a tag here, right? And a tag is a gene, is a mechanism, very simple. So what we did is, all you need to do is just to create homogeneous groups. and then see what kind of tags, what kind of genes they have. And that's what we've been doing. So this is again what I show you. So here, the very first question, which is very simple. You need to partition this landscape in groups. Now, you could do it using fancy machine learning, artificial intelligence. In our case, we use we use technical machine learning. K-means, I'm told it's machine learning, very primitive. We use K-means, but in the end, the idea here is, if I want to test my hypothesis, I want to make sure that this is really robust. So the very least thing I can do is to cut this in two and test whether red is different from blue. Because if not even red and blue are different, I think we can really drop this story, okay? These must be different. They're producing totally opposite things. And that's what we did. We partitioned this with the support of K-means, if you like, but in these two groups. And then we can produce like an average connectivity map, which is a connectivity prior. It will come, there's no need to focus on this, but these are basically regions where that let's call them like hotspots of disconnection related to all these mutations and this. And these are going to be coming, they're going to be coming useful later. But back to my question, are the mechanisms related to red different from blue? How can we test them? That's easy. You can create like in silico, a gene interactome. So you put together all these genes here, the tags, right? These ones, you can see my pointer? Yes. You can put these ones in silico with a computer, you create protein-to-protein interaction, and then you decompose them. And to cut a long story short, if you see at the blue, what happens then in terms of pathways with the blue genes and pathways with red genes, so hypoconnectivity, we call it hyperconnectivity, you see they're clearly dissociable. Blue, so out of sync connectivity, is associated with synaptic signaling, red with two major mechanisms, transcriptional regulation to some extent, and immune system dysregulation, which is very cool, because these are dissociable as expected, right? So maybe in the future, when we have, instead of 20 mouse models, we'll have 200, we can maybe split these two as well. But so far, so good. Now, you are skeptical, and I agree with you, because mice do not have autism. I'm telling you, I've told you twice. Mice can be used to test mechanisms. And so the question is, does any of this matter for human autism, right? Because that's the question. I mean, we don't do veterinary research on mice. And so what we say, okay, can we identify a similar sort of situation in human autism? And here enters the translatability of fMRI. We use fMRI in the mouse because to some extent, with 1 million caveats, we can somehow translate changes across species. So this is where we are so far. It's a summary of what I've been showing you. Different models, fMRI, we created two groups, and then we decoded them in terms of pathways. And what we did next is this. So I told you there are these big databases there and whose like decoding is so far untapped. Okay, you can download them. It's called Abide. And I said huge databases. This is 1,000 typically developed people and around 1,000 autistic people. And what we did is this. We use our mouse priors, the maps I showed you before, to guide the selection of scans in humans, if any, that have changes in connectivity with respect to control population in the very same regions of the mouse. That's nice and simple, because in mice, all the regions that mice have are also present in human brain, because the mouse is like evolutionarily old. It's clearly not the other way around. So we use this to guide, okay? It's like some sort of like supervision, I don't know. I was going to say cherry picking, but picking the brains that have this. And then we can do some sort of gene decoding. That's what we've been doing. And first of all, we do find such scans in autistic people. And here we have like, two examples, okay? That have like this sort of characteristics, which is nice because, it's not granted. I mean, it could have been like otherwise. And what is also nice is that these two subtypes, hyper and hypoconnectivity, exist throughout all the imaging scans that we accrued. There are so many, you can see. And look at the numbers, it's big numbers. And of course, these are also, if you use like discovery replication, they're highly reproducible, okay? Here, discovery data. So if you put this together, we are in a position where we have pretty much 30 to 35 people, of autistic people, that fall into these two sort of subtypes, which is cool, because we started from mice and we got this. These two subtypes are not like the mirror of one another. That's an important question. Yeah, they're pretty, widespread. But if you look like at the cognitive map, they're kind of different. Okay, and they're also slightly. I don't want to make a big story out of this, but clinically they're also slightly different. So red, so hyperconnectivity is slightly more severe than blue. But what about mechanisms? Because here this was about mechanisms. It's about proving or postulating this idea that this heterogeneity we see in humans is based by different molecular mechanisms. Okay, so what can we do here? Well, in humans we cannot unfortunately do like RNA-seq, postmortem stuff. These scans are anonymized. But what we can do it's a very simple gene decoding approach. So there's this resource. It's called Allen Institute Postmortem Gene Expression Atlas. You might have heard of this. So basically you have like this gene expression map for pretty much all the 20,000 genes in the brain. And you can, basically what we did, we took like the two average maps and we compiled a list of all the genes that are enriched spatially in the regions that are hyperconnected or hypoconnected. This way you can get two lists of genes. And in the blue we decoded 3,800, in red 4,400. What's important is these lists are significantly enriched for genes that are dysregulated in autism, which is nice check. But what is more interesting is this. When we decode them in terms of ontology, in terms of signaling pathway, we find something interesting. Hyperconnectivity is associated with this, synaptic signaling, and hyperconnectivity with immune system dysregulation, which, to refresh your memory, is exactly what I showed you before. When we just correlate mouse data to humans, these are just the mechanisms that are the most represented in the blue ones, so hypoconnectivity, and these ones are the ones that are most represented in humans and mice in hyperconnectivity, is mostly immune system. And so we believe this is an interesting result, perhaps even important results, because first of all, it shows that we were able just to start from like some sort of, if I may use this term, quote, unquote, ground truth work in mice to guide a decomposition of clinically relevant, in terms of pathology, of course, pathological mechanisms, subtypes in humans. And it also suggests that a lot of this inability of identifying this elusive, signature of autism is in fact attributable to the fact that the ethiopathology of autism is really heterogeneous. But now we have a way to decompose it, okay? And of course, imperfect, however you want, but conceptually perhaps interesting. And so just to sum up, under synchrony here, that's what I call, which is basically under connectivity or hypoconnectivity, you're expert by now, seems to be associated with synaptic alterations and this hypersynchrony, hyperconnectivity, immune-related genes. And this accounts around for 35% of autistic cases, which I think it's okay. You may wonder what about the rest, and now ask me later, I'll tell you at the end of the talk, but it's already a step forward. Now, okay, I have 5 minutes or so, and I want to move forward to the second part of my talk. Because, okay, so far so good, we kind of like, linked putatively some molecular dysfunction to what we see in brain scans. But what about the physiology? What's happening here? What does under-synchrony mean? What does over-synchrony mean? Well, one of the perks, probably the only one, of using like rodents and stuff is that you can test causally some hypotheses. And then we said, okay, let's start to investigate what's happening here. The thing is, starting from this plot I showed you before, where do we begin? There are so many pathways. Well, we started here, actually. Blue, so hyperconnectivity, is related to synaptic signaling. And we started here because if you know a little bit about these developmental disorders, when I say autism and synaptic signaling, people in the field think of one thing. Synaptic dysfunction in autism means mostly, not exclusively, but mostly excitation inhibition imbalance. Why is that? Because autism is strongly comorbid with epilepsy. 1/3 of autistic children develop also epilepsy. So we said, okay, let's start there. And this is something that we can test causally in mice. How can we do that? Well, there's this thing, It's chemogenetics. I'm not going to explain. You have to trust my word. It's a little bit like optogenetics. Probably optogenetics, everyone kind of know what it is. But instead of using lights to introduce, to switch on or switch off neurons, you use these synthetic receptors that can dial up or down the excitability of neurons. And so we said, okay, let's begin to manipulate. We tried the medial prefrontal cortex in the mouse, because we like it. And so let's try to do two things. If I said E to I imbalance, right, this may mean two things. Increasing excitation, so exciting pyramidal neurons, or decreasing inhibition. In this case, we inhibited parvalbumin GABAergic neurons. So these are like fast spiking neurons. And then we can measure connectivity. And as I like to say when I present this slide, this is really my career, the first time that my hypothesis was correct. Because really, the very first time. Because, wow, okay, this one is really like, a sanity check. Yeah, electrophysiology, you see the neurons are firing more, as you would expect. Well, population activity, okay? This is multi-unit activity. And when you look, connectivity is, as predicted, so nice, for once, blue, okay? So lower connectivity if you increase excitability. And this is an interesting result because it also comes with another prediction. If when I increase excitability, I have lower connectivity, and you might be wondering why, but I'll get to that very soon. If that's the case, when I suppress activity, so reduce excitability, I should have more connectivity, more synchrony. And that's easy to show because it's already published. We have the data in the lab already. Okay, this is again electrophysiology, what should be used basically the opposite manipulation. So multi-unit activity, neurons are firing less, and check what happens. The long coveted red is appearing. As you can see, the signal is going up over time. Cool, but difficult to explain, right? Why is that? What's happening here? Well, in order to explain, and I'm almost through, two more slides. This is very important. I'm going to explain very easily. We can use electrophysiology. We can use this is the manipulated region. I call it PFC, but it's medial prefrontal cortex. And this is like a region that is like a posterior cingulate in the mouse, so a region that is strongly innervated by the prefrontal cortex. We stuck two electrodes there. So this is not fMRI, and then we measure this. What is this? This is coherency modulation. So coherency is like correlation between signals. Think of fMRI connectivity, in this case with LFP. But given that LFP local field potential, you think it's like an EEG. So here, this is like the interesting thing about LFP or EEG is that you can do it across frequencies, right? You can see here. So this is gamma, whatever, yeah, theta, and so on. And what you see here is the connectivity with the electrophysiological profile produced by our manipulation. So red is when increase excitation, and blue when we reduce inhibition. And you can see they are very different, right? They're very different. Yet, in my fMRI maps, they were both blue. Still, here they're very different, okay? And so you may be wondering, what the hell? What's going on here? Well, in order to account for this, you have to remember an important point, that fMRI is a very slow rhythm. So maybe what we should focus our attention on is here. And I'm going to help you. This is, again, replotting on a log scale. And you see what's happening here. On this infra-slow activity, both manipulations are below zero. Zero means that they are not different from control. So if they are below zero, means that they are out of sync, okay? They are lower coherency. And once again, this is interesting because if this is true, then this comes with a prediction that when I do the opposite manipulation, I reduce excitation inhibition balance, I should have more coherency here, irrespective of what's happening here. And that's what we got. If I throw in also that electrophysiology, you see in this very slow band, infra-slow, I have stronger connectivity. And the last prediction, and I'm going to wrap this up, is that if this is really important for fMRI, the magnitude of this coupling at a very slow frequency, below 1 Hertz, should covary with the amplitude of fMRI changes that are produced by these manipulations. And when we just plot them all together, we see this is the case. And this is the case only for slow fluctuations, not for slow rhythms, and not for faster rhythms. Okay, it's a lot of stuff, but I'm going to sum up in the last slide, which is this. Out of this, we can learn a general rule. Is it written here? No, I've forgotten it. Anyway, listen to me then. The rule is less is more and more is less, okay? So fewer, multi-unit activity, lower excitability, produce more connectivity. Why? Because you have these low signals that are not desynchronized by the activity of a single region. And the other way around, it's reported here, okay? And this holds only if you consider this postulate, which is, once again, it's a postulate, but it's not so surprising, right? fMRI connectivity is dominated by changes in very slow neuronal coherence, okay, neuronal rhythms. So the faster rhythms are important insofar they impact on the slow rhythms. Okay, and so basically this is what I was going to say. Neocortical excitability, so total firing activity, is inversely related to fMRI connectivity. And so, getting back to my original question, when I show you this could be synaptic alterations, but this may indicate increased excitability, right? Increase it to high balance. And hyperconnectivity, which is related to immune-related genes, may indicate reduced, well, excessive inhibition, so to speak. And so, that's where we are, and I'm going to wrap up because my time is up. And this is clearly like a bit of a simplification. I'm sure it's more nuanced than that. But what is nice that this stuff is clinically testable, and we are testing it, via collaborations. So, my talk is over. I am really to thank all the super talented people that contributed to this sort of like work that lasted more than 10 years. We are also hiring, someone else here, if you guys are interested, please send over people to my lab. Thank you so much.
Thank you, Alessandro. Are there any questions in the audience? Don't be shy. Mercè, please.
Hi, thank you for the super nice talk. It's quite interesting. I will focus on the first part of the talk where you talk about these different networks according to plasticity or to inflammation or more immune response. Do you think that this applies to any disorder of the brain, so that we have the two types of...
Very much so.
That was a clear answer.
I mean, I can elaborate, of course. Yes, very much so. Once again, with all the limits of this model, which is corticocentric, I don't think it applies to hippocampus, other regions that have stronger rhythms that they can be like nested. But yes, and we are putting together also a perspective on this. For example, dementia. It's interesting that people for many years have been scratching their heads about the fact that with dementia in the early stages, you have this increased connectivity. Why? And there are also reviews. They say, oh, maybe the brain is like overcompensating with a longer path. But the very simple explanation is this. You are beginning to have neuronal loss, and then, cortical excitability, you have like lower input locally, and then fMRI connectivity is taken over by these very slow, less localized oscillations. And so the brain is over-synchronous. And this makes a lot of sense because in Alzheimer's, very often you have more delta activity, stroke the same. In the stroke, in the ipsilateral region, you have more delta. And and then you have more connectivity. This is, I think, a very simple model that explains a lot of this. Not everything, I don't want to be arrogant, and it has to be tested. But I think yes, it's a sound yes, my answer here.
Thank you.
Very interesting talk. So since one of the pathways that you have more altered, that is mTOR, for example, well, maybe in the mouse models, of course. No?
mTOR, you mentioned?
Yes.
Yes.
So did you try to treat pharmacologically with rapamycin and revert this?
We did exactly that. It's published in 2021. The mTOR study, it's a very beautiful. It's a study I like a lot because, yes, we did exactly what you suggested. Those mice have more spines, they have more dendritic spines, they have more connectivity. And you may wonder why. Well, because once again, spines are linear integrators of these low oscillations, right? They are addictive. If the spines are functional. We tested that they are functional, they are more than necessary, they have more connectivity. You give rapamycin, and then you normalize both spines and connectivity. So that's also interesting because I think, yeah, dendritic spines may play a role at the microscale level.
But, well, rapamycin inhibits mTOR, but not all the activities of the mTOR. So maybe you have
of course.
Some activities that you are not inactivating there. So, but it's really
of
interesting.
Of course. I mean, we linked it to spines. And we have other data, but I don't want to monopolize this. Absolutely. Maybe we can discuss offline. Yeah.
Okay, nice. Thank you.
Hi, thank you. Very nice talk. I found really interesting this relationship between the connectivity and the excitability. And in this model, from what I understood, you did the manipulation and you get it from the beginning, from birth? No.
Nope.
And I was, well, my question was regarding...
That is a separate, super cool question.
Well, sorry, just to finish, but what I was wondering is that, depending on that, but also if then you would expect different results depending on the time point where you do the modification, and if you would expect different... Yeah.
That's a great question, because that's exactly the question we had in the lab. So these manipulations are, we call them acute, in adulthood. Switch on, switch off. And you say, okay, but for example, autism is developmental, right? So for that one, this is an important question. We generated a mouse line that overexpresses these dreads postnatally. We treat them, and I haven't reported it here. It's unpublished, but we have a cool story. Long story short, very long but short, is that if you do that, at the population level, mice never recover. So if you increase excitability in the early postnatal window, those guys have like EEG alterations that are indicative of excitation inhibition balance and hypoconnectivity. So that's another piece of the puzzle that, suggests that maybe we are onto something.
Thank you. I have questions, but I will ask you later because we need to move on to the next speaker, Dr. Albert Giralt. He's Albert Giralt, Associate Professor of the Faculty of Medicine and Health Science in the Department of Biomedicine. He was Ramon y Cajal, a researcher, and he's the head of the Neuroplasticity, Neuropsychiatry, and Neurologic Disorders Group. Welcome, and whenever you want.
Okay, thank you very much, Lupe, for this very nice introduction. I appreciate it, and also thank you very much to the organizers for inviting me today to give a talk and to talk about the main lines of research in our lab in this phenomenal and wonderful multi-brain congress. And I also thought that it's super interesting that you put my talk in this mini symposium called Challenges in the Pathological Brain, because in our research, the people that work with psychiatric disorders, we usually have to deal with huge amounts of heterogeneity in the samples or subjects, or we are working with multifactorial disorders, and we have to deal with many things and stuff that sometimes you cannot control, right? And in our lab, we strongly believe that one way to address or to overcome this kind of variability and this complexity of these psychiatric disorders is by using translational approaches. So that's why today I'm going to talk about translational approaches to understand schizophrenia in this context. So my talk is divided into main parts. In the first part, I will talk about the aberrant interactions between the immune system and the central nervous system by using translational models in the context of schizophrenia. And in the second part, I will talk about how we are characterizing and profiling the changes in small RNAs in a particular brain region in the context of schizophrenia too. So going to the first part of the talk, let me put you a little bit in the context. So as I told you before, and I think it's clear enough, psychiatric disorders like schizophrenia have several heterogeneous symptoms, like positive symptoms like delusions and hallucinations, or negative symptoms such as deficits in sociability, or also cognitive symptoms such as attentional problems or working memory alterations. And also, to make the story more complex, this disorder is multifactorial. So there have been described a huge and a substantial role of many, many risk factors, like, for example, environmental risk factors, habits, genetic risk factors, or chronic stress per se, and finally, immunological challenges. So in this context, nowadays, there is an intense body of research focusing on the interaction between the immune challenges combined with genetic risk factors and how do they interact in order to induce, to provoke the development of the disorder, right? And we are in this context. We are working in this topic. We are focusing with these immune challenges. So in our lab, we have the following hypothesis. We strongly believe, or we hypothesize, that in circulating immunocompetent cells, like, for example, lymphocytes, they are affected by these kind of combinations of risk factors, for example, genetic risk factors or environmental risk factors involving immune challenges. And in these cells, this combination of factors induce alterations on gene transcription. In turn, these alterations in gene transcription will alter the profile of the released molecules, what we call secretome. These molecules include cytokines, chemokines, growth factors, these kind of molecules that are super involved with inflammation in general and also with neuroinflammation. In this hypothesis, we think that this aberrant secretome is able to reach the brain and to exert an impact in specific and relevant neural networks that are involved in the physiopathology of schizophrenia. So this is the context in which we are working now. But the idea is, okay, I started the talk talking about the relevance of the use of translational models to understand complex disorders, right? So how to do that? So first of all, we have to collaborate with clinicians, of course. We have strongly collaborated with Dr. Arranz. She's a psychiatrist and consultant and a principal investigator in Hospital Sant Joan de Déu. And together we have been able to recruit and to collect some blood samples from several patients with schizophrenia in one side, and on the other side, several blood samples from control patients without schizophrenia. From these patients and from these blood samples, we have isolated PBMCs, peripheral blood mononuclear cells, that are circulating immune cells. Then we have cultured them, and in culture, we stimulated them in order to get an enriched secretome to work with it. So the secretome is collected, which is the superneuron, after a centrifuge, and what we can do with that, right? So what we can do, first of all, is to profile it. And we can do it in several ways. The first way that we did is just a common method by mass spectrometry. We profiled the secretomes from patients with schizophrenia and controls, and we identified several dysregulated proteins. So what? It's a profile, it's nice, very fancy. We have several potential targets, but what we do with this, no? So the first step is to validate, to test whether this aberrant secretome is provoking something. Is it biological relevant? How to do that? We have combined in vitro techniques and in vivo techniques. First, I will explain you an in vitro technique. Again, in collaboration with Dr. Rabadán, she's a CEO in the xeneuroids. We have generated in collaboration this kind of primary neurospheroids. These are clusterized primary neurons from mice, and we have transduced them with adeno-associated virus expressing GCAMs, and this is the scheme of the experimental approach. No? We have clusters of cells that can be easily monitored. So, and which is the experimental design at the end? So we are using these superneurons or secretomes coming from cultured PBMCs from patients and controls, and we are transferring them to these monads. These cells, these clusterized neurons are called monads. And these monads, what we can do with them? We can monitor the neural activity of these ensembles, of these clusterized neurospheroids. So as you can see here in the video, is a pretty easy way to analyze several parameters. For example, neural firing rate, the intensity of the firing frequency, and also the synchrony, which is super important. And actually, here in the right panel, I'm showing one of the most interesting results from this experiment, which is evaluated by using average pairwise correlation. And as you can see here, this is a measure of neural synchrony. And you can see that in neurospheroids treated with secretomes from patients with schizophrenia, this coordination, this synchrony is reduced. And this is super exciting because we think it's exciting, of course, because it's a measure, well, it's what occurs in schizophrenia, a reduction in neural synchrony. Okay, now we have a readout on neural networks activity, and is that playing a role in high-order cognitive functions? So let's test it, and how we can test it. Again, we have used these superneurons, these secretomes from cultured PBMCs from human patients, and in this time what we have done is to transfer them or to transduce them in vivo in mice. What we have done is using mini-osmotic pumps, and these osmotic pumps, we put the secretome, the human secretome in these pumps, in a conditioned media, and this is progressively released into the lateral ventricle. And then this is made, this protocol or this procedure lasts for several days, and we can evaluate several behavioral phenotypes. And I will put you just one example. I told you at the beginning of the talk that social deficits are among the most important alterations in the symptomatology of schizophrenia, and we have evaluated this by using a simple paradigm, which is called three-chamber social interaction test, in which mice prefer to socialize with a strange mouse than compared to an empty cage. And these have been the results. In black, you have the control mice that are receiving control secretomes from control patients, and they prefer to explore or to socially interact with a stranger mouse compared to an empty cage. In the case of mice that are receiving this secretome from patients with schizophrenia, we have no preference. So these mice are developing somehow social deficits as it occurs in schizophrenia. So that's the first part of the talk. So we have been able to perform or to generate, we think, very translatable models for schizophrenia in terms of neuroimmune interactions. And now let me talk about a little bit the second part of the talk, a little bit longer, and in which I will talk about how we have profiled these changes on small RNAs in the hippocampus of schizophrenic patients, and we have evaluated if this is playing a role in cognition in translational models. Just to put you a little bit in context, but just a little bit because I'm not an expert on small RNAs. Small RNAs are non-coding RNA sequences, and there are several species with different potential functions, but in general, what it has been described is that these small RNAs play a role in gene transcription regulation, and mostly through inhibition of gene expression. Actually, it has been postulated that 33% of the total genes, or the genome, can be regulated by these small RNAs, so a big readout, a big frame. And what about small RNAs in schizophrenia? This is a hot topic nowadays in the field of schizophrenia because during the last months, some have raised up some seminal papers in very important journals like Science Advances, in which the authors have shown or they have characterized differentially expressed small RNAs in circulating extracellular vesicles or in other peripheral tissues. In these papers, in most of them, they have performed a huge analysis, huge amount of samples, huge number of patients, and super fancy statistical tools, blah, blah, blah. But still, all of them are descriptive. So what we are trying to do is go a step beyond, to try to evaluate which is the role of these changes on small RNAs. So what we are doing now, in collaboration again with an expert in the field, Dr. Eulalia Martí, she has been able also to previously publish a similar kind of work, mimicking Huntington disease profiles or phenotypes by introducing in the brain of mice, in the mice brain, isolated human small RNAs from Huntington disease patients. And what we are doing right now is mostly the same. So what we are doing is we are isolating small RNAs from the hippocampus of control patients. And on the other hand, we are isolating small RNAs from patients with schizophrenia. In this part of the talk, in this project, keep in mind that we have two poles, as you can see here, and why we did that. In this project, we have considered the heterogeneity of the disorder. So the reason why we have used two poles is because we are going to focus on the changes that are occurring in both poles, because you will see some examples that there are several changes that occur or are happening in one pool but not in the other one. So it's a way to filter the results and to focus with the relevant ones, you will see. And again, for this work, we have to collaborate with clinicians, again, with Dr. Belén Arranz, the psychiatrist, and which kind of readouts we have observed or identified by doing these kind of experiments. The first all is, again, we have to profile the human samples. And as you can see here, these two volcano plots, we are comparing the pool 1 of schizophrenic patients versus control, and the pool 2 of schizophrenic patients versus the control again. And okay, this is a very nice graph, several differentially expressed small RNAs, but I prefer that rather you take a look to the table at right. Here you can see in a summary the small RNAs that are changing only in the pool 1 and the small RNAs that are changing only in the pool 2, that are several, but we have decided to focus with the changes that are occurring in the same line, in the same sense, in both pools. So these upregulated small RNAs that are co-deregulated, let's say, in both pools. In summary, we can say that there are up to 8 small RNAs that are similarly changing in both pools. So we think that this is a way to focus with the detected changes. But again, the question remains, which is the role of these differentially expressed small RNAs? So we have described a plethora, a myriad of changes, but are they playing a role in the physiopathology of schizophrenia? Let's test it. So what we have done here is to use wild-type mice, and we have generated 4 groups. The 3 first groups are these 3 groups of mice. The first group of mice, the control mice, are receiving isolated small RNAs from control patients in the hippocampus. The second group of mice are receiving isolated small RNAs from the first pool of schizophrenic patients, and the same for the pool 2, or the group number 2, that are receiving isolated small RNAs from the second pool of patients with schizophrenia. In this experiment, we have added a new control, which are the mice that are receiving just vehicle, PBS, I don't remember. And this is just to control whether the injection of small RNAs per se is able to induce some detrimental phenotypes, some aberrant phenotypes or some affectations. It's an additional control. So, as I told you, well, we have isolated and profiled small RNAs from the human hippocampus, and we have injected these small RNAs, hippocampus small RNAs, to the mouse hippocampus. Then we have to evaluate hippocampal-related functions. And I will show you just one example. We have evaluated spatial short-term memory in a T maze. In this maze, we are evaluating the preference of mice that the mice show to explore a new context compared to an old one. And here you see the results. In both cases, in both controls, the mice prefer, in black, the vehicle ones, and in blue, the mice that received small RNAs from controls. Both of them are they prefer to explore the new context, so normal spatial short-term memory. And in contrast, we can see that mice receiving isolated small RNAs from the 2 pools of patients with schizophrenia, they do not show any kind of preference for any context. So short-term spatial memory deficits. To go further in detail in this, we try to look for neuronal correlates in terms of neuropathology, and we evaluated dendritic spine density in the pyramidal cells of the CA1. And again, we observed that there is a reduction on spine density in both groups of mice receiving the small RNAs from pool 1 and pool 2. Both of them have, you can see here, a clear reduction on spine density compared to the 2 controls. Well, so the results, we were super happy when we observed this kind of strong results and the coherency of these results, because both phenotypes, this kind of phenotypes are commonly observed in schizophrenia too. So in post-mortem samples with schizophrenia, a reduction in spine density has been shown, and also a deficit in spatial short-term memory is a common cognitive deficit in patients with schizophrenia. So we were super happy with the results. And then we were into further detail exploring which is going on in this hippocampus from these injected mice, right? So what we did in this case is to perform RNA sequencing, bulk RNA sequencing from these injected hippocampi from these mice. And you can see here these very nice volcanoplots. Several genes are upregulated in red or downregulated in blue in the two comparisons. But again, in this case, since we have a huge amount of variability, it's better to look at this table. As you can see here, there are several genes that change in pool 1 and several genes that are changing in pool 2, but only 77 are changing in the same way and are upregulated in both of them compared to controls. So we have to, in our opinion, this is a super filter, and we are focusing with these 77 genes in the case of upregulated genes. And also there were only 56 downregulated genes that were commonly downregulated in both pools compared to controls. And you can see here more than 100 and 400 genes. This is massive. The variability is super big. So we were super happy then when we realized this kind of variability because we have took this decision. And finally, what we did is to deepen on these profiles. And we conclude that by using pathway analysis, that mostly synaptic alterations were the most affected by these commonly deregulated genes. For example, pathways related with vesicle synaptic transmission, synaptic signaling, or vesicle turnover, this kind of stuff. And now we are working with that. So this is what I wanted to tell you. I'm on time, no? Perfectly on time. So, congrats. And the two take-home messages could be, well, I think there are three take-home messages. The first one is that the aberrant immune secretome impairs neural circuits and induces schizophrenic-like symptoms, such as deficient social skills. And the second take-home message is that altered small RNAs composition may be sufficient to induce specific cognitive symptoms in schizophrenia. Right now, well, we have much more results, but I didn't want to explain everything. But what we are doing right now is to go in further detail, trying to explore the specific engrams and circuits involved with these deficits in both projects, and also the underlying molecular mechanisms. We are working on that super hard. And the third take-home message could be maybe that translational models are super useful, in our opinion, in the field of psychiatric disorders. So nothing else, just my acknowledgements. Many, many thanks to my team. I have to say that the project about small RNAs is being led by Marcos Galan, and the project regarding to the neuroimmune alterations is led by Ivan Ballask and the rest of the team that are doing also super interesting projects. Also, I would like to thank to the major collaborators. Without them, nothing is possible. And of course, the funding agencies and sponsors, and you can find me in the social media, in Twitter or X, in this account. Thank you very much.
Thank you, Albert. You were perfect on time. You always show very cool things, and I think that these models are really, really important. Are there any questions in the audience? Yes.
Great talk, thank you.
Thank you.
I have a couple of questions, try to keep them quick. So the first question is the secretome one. Is what you injected pro-inflammatory, and in such case, did you consider using like LPS as a control? Because there's evidence, right? If you inject LPS intraventricularly, so can you speak to that?
Yeah. Well, we have talked, because the time was short, but we have profiled this secretome, and before that, we have characterized if this secretome is toxic per se in control conditions. So we have to set up the system, because depending on the dose, just adding secretome in cells is toxic. So we have to adjust the dose. I don't remember the dose now, sorry. Yeah. But it's very low in order to have available neurons and not unhealthy mice. And the second part is regarding to the molecular profile. We had changes in both directions, changes in pro-inflammatory molecules and changes in anti-inflammatory molecules, but most of them were in the direction of pro-inflammation. Because, for example, a reduction in interleukin-4, I think, is anti-inflammatory in general, and an increase in, I don't remember the cytokine, pro-inflammatory cytokine, yes.
Okay, and the quick comment I have also in the same study, when you measure sociability, be prepared to receive the question of measuring interaction between conditions, because the fact that one is significant and the other is not, is statistically not robust enough. I mean, reviewers might ask you, so just it's a suggestion for you to go over that specific testing. I explain later.
Okay.
But I didn't want to forget that this is important.
Well, we perform a comprehensive behavioral analysis, including several parameters, cognition, sociability, and agitation, and this kind of stuff. All of them are rudimentary for mouse models, we understand it, but yeah, we can discuss further about that later.
Thank you.
There's another question there.
Thank you for your talk. I was just wondering if you checked if there is any correlation between the small RNAs upregulation that you observe in patients and the upregulations in genes that you observe in the hippocampus of mice.
In the mouse hippocampus?
Yes.
Yeah, Marcos will be happy about this question. I don't know if it's in the audience. Well, yes, we had many coitus interruptus because we observed many proteins that in one pool were the same, were differentially expressed compared to mice, but other ones were not. But we have focused in a couple of proteins that seem to be like there. I think there is, for example, let me be clear. There are a protein involved in synaptic transmission in interneurons that is similarly deregulated in mice and in one of the pools of the human samples. But in the other pool, we have not these results. So we're struggling with that, and it's not easy to find a perfect correlation between the human samples and the mouse samples. That's the heterogeneity that I was talking about before. But yes, it's a super interesting question. Thank you.
Thank you.
I have a question, Albert. Me.
You. I need water.
Maybe you have to tell me again. I don't know if I already asked you this. Pool 1 and Pool 2 were different kind of patients, like with different symptoms or not? They were all...
All of them shared, even though the RIN levels, the mean quality controls were similar, and the treatments were more or less the same, the distribution of the treatments, clozapine or whatever, were the same. The only bias here was that maybe the post-mortem interval was a little bit different compared to controls, not compared between them. Between them were equal.
Okay. So, thank you. Thank you very much. Oh, there's another question.
Hi, thank you. I was actually curious about this, of how you distinguished or dissociated the two pools, if there was like a clustering analysis or what other variable they were based on.
But can you repeat the question?
How you dissociated the two pools or distinguished them from one another.
How we distinguish them? What do you mean? Well, we try to perform similar groups, sharing in terms of postmortem time interval, the age, and also the treatment. So they are equal in many terms.
I guess, I mean, like, how did you dissociate? How did you create the two groups? Was there a clustering analysis or?
No, the only way in which we clusterized the groups was in terms of the same age, the same age average, the same postmortem interval, and the same RIN values.
Okay, thank you.
We have to move on. Last very quick question.
Hi, Albert. Thank you for your talk. Regarding the secretome, I have a question. Have you tried the secretome of other psychiatric conditions also related to immunologic, related with the immune system? I mean, how specific is the secretome in this condition for schizophrenia or for depression? Because then the phenotypes, the symptoms, some are common in different psychiatric conditions. So I just wanted to know if there's a specificity in the secretome for any psychiatric condition or not?
Well, maybe it's a spoiler, but we are working on that. So we are collecting samples from other psychiatric disorders, and a potential project idea is to compare between them, if they share some molecular signatures or not, and then to test them in these models, in these translational models. But I don't know if I'm allowed to say much more than this.
Very, very last question.
Sure, yeah, thanks for the talk. So I was wondering, you comparing the two models, social dysfunction and short-term memory. So is short-term memory affected in the secretome model and social interactions affected? So are these behavioral phenotypes specific of each model?
No. Well, let me see. First, in the model in which we injected the small RNAs, we injected the hippocampus. So what we evaluated was mostly hippocampal-related tests. But I have to say that we only evaluated anxiety or locomotion and declarative memory is not so related with the hippocampal function, and they were preserved. So in this model, it was pretty specific on a hippocampal-dependent function. In the model of the secretome, since we are injecting the lateral ventricle, we are reaching almost the entire central nervous system, and we observed deficits mostly in sociability, but also in some declarative memories, but not in agitation or positive-like symptoms.
Thank you. Thank you. Congratulations, Albert. So we move on to the next speaker, and it's also my pleasure to present you to Judith Dominguez-Borràs. She is also Associate Professor of the Faculty of Psychology and Independent Researcher at the Brain Lab. Since last year, she holds also an ERC starting grant, and sorry I have to say this, but she was nominated by RAC-U, the radio station, as one of the 7 Catalan scientists you need to know. So it is our pleasure to have her here today.
Well, thanks, Lupe. Thanks a lot. Thanks a lot for inviting me. I have to say I am deeply surprised to be part of this so interesting symposium. What I'm going to talk about now is completely different. Might be some anatomical relation, some anatomical link to what has been said just now in the previous talk. So we will move from the hippocampus to somewhere nearby, the amygdala. But we will definitely change the scale. We will zoom out, and we will move into human cognition and human research. So I'm going to talk about, I've always been interested in the study of emotion processing in humans, basically, the neural basis of emotion processing. And this is a very specific thing. What I'm going to present is what we are currently working on. So cortical emotion processing, but specifically in the human auditory system. So I am also from here, from the University of Barcelona. I am based at the Faculty of Psychology. And I will try to be brief. So this is based on a phenomenon that, so I will just put you in context. We all know that survival depends deeply on us being able to efficiently process and efficiently respond to affectively salient stimuli. So anything that has an affective content is more readily detected by us. And we have seen this in humans behaviorally. So we tend to detect better and to process, centrally speaking, better the stimuli when they are emotional in respect to when they are not emotional. And this usually correlates at the neural level I'm just giving you a spoiler. Can I go back? So here. So this is the pointer. So this is a very well-known phenomenon. So whenever we are visualizing a fearful face versus a neutral face, there is always an increase. This is just the fMRI data showing that there is a sensory increase in the neural response, and which is specifically in selective regions that are specialized in processing a specific category. So in this case, it would be an increase in fusiform gyros for fearful faces versus neutral faces. And this is just another example of an increased response with intracranial EEG in epileptic patients implanted directly on the fusiform gyros. So, and this is just showing us that this increase is rather early, around 200 milliseconds or earlier post-stimulus onset. So this is a very, very typical phenomenon. And we see this across sensory modalities. We have also seen it. So for example, if we process emotional voices versus neutral voices, we can see an increase again in voice areas. In the superior temporal sulcus or gyros, but at the same time, so this not only happens in association areas, but also in primary sensory areas. For example, this is just another random example of another study where we saw an increase in primary or secondary somatosensory regions when subjects were processing some touch stimulation on the cheeks, and these stimulations were associated to emotion somehow. So this is a very well-known phenomenon, and if we talk about this phenomenon, we need to talk about the amygdala. So the amygdala, this has been seen in humans, but also in animal literature, is a structure that has a very important role, of course, as everybody knows, in emotion processing, but it has a number of, its role is very, very widespread. So it has an enormous, different roles in many aspects of social cognition, but more basically, it is related to salience detection. So it is directly involved in the phenomenon that when we visualize or we process an emotional stimulus, we increase the sensory response, and it somehow seems to boost awareness of the stimuli. You will have to believe that, because I'm not going to show direct evidence for boosting awareness, but at least we know, for example, from evidence in humans that amygdala lesion suppresses this increased sensory response, this typical phenomenon. So whenever patients or healthy controls need to process emotional pictures versus neutral pictures, this typical increase in the sensory cortices that we observe in healthy controls disappears ipsilaterally to the side of the lesion whenever there is an amygdala lesion. So this would be suggesting that the amygdala has some important role on that, and it even correlates with the amount of, the degree of amygdala lesion. At the behavioral level, we also have some evidence. Whenever we have this would be a single patient with bilateral, very extensive amygdala lesion. Well, we did many tests with this patient, and we observed that consistently the patient was showing an impairment in the detection of emotional versus neutral faces in this typical visual search task. So there is an impairment not only at the neural level, but also at the behavioral level. More specifically, this is just the last thing to put you in context on what we want to study. The amygdala might have a more directly implication in this boosting of stimulus awareness when we encounter emotional stimulation. We have some data, some relatively recent data from intracranial patients, intracranial EEG in epileptic patients implanted on their amygdala, where we presented some very a task that masks very strongly the stimuli. So the stimuli can become either subliminal or they can remain supraliminar. And what we have seen is at least a correlational relationship, not necessarily a causality, but at least that activity of the amygdala before the stimulus correlates with subsequent detection of the stimulus. So it might have a more direct involvement in this boosting of awareness. But there's other evidence in patients, but I will not go into details. But this was just to say that the amygdala is known to modulate perception via different outputs. It's virtually connected with the whole brain at the cortical and at the subcortical level, not only in humans, but I mean across species. But we know that it sends projections to virtually all sensory systems. It is very well connected to primary and association cortices across all sensory systems, but also indirectly the amygdala can send projections to the attention systems, cholinergic systems, noradrenergic systems. But the question here, and now moving into what we are doing now, an overview, how does the amygdala receive this sensory input? So how do the sensory systems send the information to the amygdala in order for it to decide whether something is emotional or not? So we know that the amygdala, and specifically the basolateral amygdala, receives inputs from a number of regions, but specifically, and we will focus on the sensory system, so both at the cortical and at the subcortical level, there is a lot of evidence that the sensory systems send some slower or some faster inputs to this structure, and then this structure can send the projections to influence perception and awareness. So specifically in humans, there is one route that has been widely studied, which is essentially visual, and I will just call it the visual subcortical route for fast emotion detection. It has many different names, but this visual subcortical route would consist of, or is believed to consist of direct inputs from the retina to the superior colliculus, and then from the superior colliculus to the pulvinar of the thalamus, and then from the pulvinar of the thalamus straight into the amygdala. And this would allow the amygdala to rapidly detect emotion and send its projections for orchestrating adaptive responses before the information would reach the cortex. So this pathway has been, this visual subcortical route has been studied widely in humans, and it is believed to be the responsible, so it has a reason to exist, so it is not vestigial. It is supposed to be responsible for fast and efficient emotion processing or emotion response, and it is believed to be specifically sensitive to very rudimentary inputs. Actually, it is part of the magnocellular visual pathway. So, and this has been proven in humans that whenever we present emotional faces in this format, like filtered, so we only get the low spatial frequencies, this is just one example, so we tend to activate structures that are involved or are part of this subcortical visual route to the amygdala. So it is believed that at least, well, or in its majority, it consists of magnocellular neurons. Also, this visual subcortical route is supposed or is believed to be present in all vertebrates, even in those species that do not have a cortical route for vision. And at the same time, some say that it might be the one that is present at birth before, so that actually babies would be able to detect emotional visual stimulation, even in the absence of a mature cortical visual system. So the question that we're trying to ask in humans is whether a similar subcortical route for emotion to the amygdala exists in humans. In addition, and why, I mean, it should exist. I mean, of course, first of all, is that all this, the visual subcortical route and the concept of the visual subcortical route in humans actually comes from animal evidence, and most of the early evidence that was described in animals was using fear conditioning, and it was focused on the auditory system. So it does exist in animals for sure, but the thing is that it is uncertain in humans, right? So that's what we are working on. So we're trying to characterize in humans such a subcortical pathway to the amygdala using different techniques. We are starting, this is our starting hypothesis. Basically, this is what it has been described in animals. We assume that there will be an MGV, so a direct connection between the auditory thalamus to the amygdala before the information would reach the cortex, and this would allow the amygdala to elicit a fast, to emit a fast response. But there are other potential pathways that we are testing. I will show you now. So our hypothesis is that, and we are trying to use many techniques. I'm just going to show you some scalp EEG and pupillary data very, very quickly, and also some tractography data. So we're trying to characterize it from different perspectives, the timing, so, and then the function of the pathway. So we are assuming that we should be able to find in humans a route that is auditory, but it is essentially very similar to the visual route. And we want to find, or we want to see, because the idea is that in the auditory system there is no magnocellular route for audition. It doesn't exist, such as the one that we know in vision. But in audition, there are magnocellular neurons that have been described in all studies in animals that are actually in, some of them are in the auditory thalamus, for example. So we expect that it should be, and actually there is sufficient data to think that both types of cells are very, very similar in terms of the way they respond, the parameters they respond to. So it should be very similar, and we are trying to find parameters that might activate this pathway. So we're trying to study it at the functional level with humans, which is difficult, but we're trying. We know, for example, this is our starting hypothesis. So we know that these neurons are very sensitive to high amplitude modulations in sounds. And this could be a good candidate to be a homolog parameter in the acoustic domain to the low spatial frequencies that were described in the visual system. So we're trying with this parameter and see what happens. I can show you very briefly some data that my PhD student Martina is currently working on. So we have some evidence that we can obtain early pupillary response to emotion, to emotional stimuli, only when these are presented at high amplitude modulations. That would be an example of a sound that has a high amplitude modulation, so rapid changes in time of amplitude, and this would be as opposed to slower or lower amplitude modulation. So this is just a parameter that we were trying. We did a very simple conditioning paradigm where we presented the sounds that were just voices filtered at different amplitude modulations. And then we see here the typical absence of response, pupillary response. This is pupil, this is pupil. Preconditioning, there is no increased response. Here there is an increased response during the conditioning. But the interesting part was that we found a very striking early response that only appeared when the emotional stimuli were at high amplitude modulations. This, of course, at first, well, it might still be an artifact, so we don't know. But the thing is that she has found that this early response correlates nicely with the early auditory response to emotion that we have also observed only for high amplitude modulations. And also at the behavioral level, in the participants that had low anxiety trait, anxiety, it correlates with detection. So this early response might have some meaning. In any case, this might suggest that we might be on the right path. By using this parameter, we might, so actually we cannot say that we are activating the subcortical route for additional auditory emotion processing, but at least we can say that using this acoustic parameter, we are obtaining some fast responses at the cortical auditory level also, but also at the peripheral level, for example, with pupillometry. On the other hand, We are also trying to characterize anatomically. At some point, we will put everything together, hopefully. But we're trying to test different tracks with tractography. And as I said, this is this is the starting hypothesis, a direct connection between MGB and amygdala, but there were other candidate pathways, for example, a direct connection between inferior colliculus and amygdala. This has been described only once in a human tractography study, but we just felt that we had to test it. And also in bats, but I think it was the other way around, amygdala to inferior colliculus, but nothing else. So it has never, ever been described and certainly not in humans. And then another hypothetical or potential pathway that we are testing is the auditory pulvinar. The pulvinar is not only very well known for being visual, but it is essentially multisensory. So, and actually, even though you can find auditory responsive neurons all over the pulvinar, it seems to have some subregions that are more likely or more prone to respond to auditory stimulation. So we also wanted to test it was anterior pulvinar, I think, at least, and some medial parts of the pulvinar. So we tested also this tract. My student, Emma, thanks to her valuable work and also with the help of our colleagues in the Laboratory of Surgical and Experimental Neuroanatomy at the University of Barcelona. We're trying to, we were able to reconstruct some of these pathways. These two seem to be the most consistent. So inferior colliculus to MGB to amygdala, and auditory pulvinar to amygdala. Emma, this was using some data from the HCP database on 200 subjects, and the interesting thing was that we could access to some scores from some emotion questionnaires, and then the density of these tracks seemed to correlate with fearfulness, some feelings of fearfulness, something that was asked in questionnaires. That was the closest we could find to emotion processing. And at the same time, both of them correlated with acoustic processing abilities, hearing ability in noise. So both of them correlated with, so these correlations indicate that at least we can say that they somehow are auditory and that they are somehow related to emotion processing, which is always encouraging and a good starting point. And finally, we are currently trying to reconstruct many other subcortical amygdala tracts, mainly stemming from the thalamus, for auditory. So we basically took all the animal literature and we have listed all the tracts that we wanted to replicate in humans, because and the ones that we thought would be mainly auditory and of course for fear processing. So connecting mainly the thalamus with the amygdala. Most of the data of course comes from rodents and from classic studies from the 80s. So this is the first one that it has been tested. This was in collaboration, this is currently in collaboration with our colleagues at the Basque Center on Cognition, Brain and Language. And it is being done on a sample of 113 subjects that was previously published recently. And this is just the image of a single subject, but this is a list of the most stable connections between the thalamus and the amygdala. So the main one is thalamus and amygdala. It seems to be, it was super robust in this sample. But also, interestingly, we could, or for now, it seems like we can reconstruct connections, direct connections from inferior colliculus to the basolateral amygdala bypassing MGB. This was specifically tested, and it seems like most of the tracks could survive. So most of them were direct, so we can say that they are direct. And we are also testing, we have tested subregion by subregion, different subregions of the pulvinar, and interestingly, all of them seem to be connected to the basolateral amygdala by showing trajectories. We cannot see it too well here, but the interesting thing was that for some reason, the anterior part, the anterior trajectories, the ones that stemmed in anterior pulvinar, seem to be independent from the others. And we still have to see why this might happen. And these were the other ones that, of course, when you work with nuclei in the thalamus, in humans, and nuclei in the thalamus in the animal literature, it's very complicated because there is no consensus in the naming and the parcellation, but we're trying to just we're trying to do a comparison, and we will try to make sense out of all of this. So these were the other most robust connections. For example, the mediodorsal thalamus showed strong connections with basolateral amygdala in humans, whereas in animals, as far as I remember, it hasn't been shown, so we don't know. Anyway, the conclusions, I hope I'm on time, is basically the amygdala acts as a salience detector, modulating the sensory systems. In vision, there is a visual subcortical pathway to the amygdala that is well known in humans, that has been widely studied, allowing for crude but fast emotion detection. A similar subcortical route may exist for audition in humans, which we are currently studying, and it may respond to similar stimulus parameters to those in the visual route, but we have to find out which ones. There are other candidates. And, distinct subcortical tracks to the amygdala for auditory fear processing have been reconstructed in humans, but we, of course, need to further characterize them, and we ideally should see whether each of them holds a specific function in human emotion processing, whether all of them are in fact auditory or visual or multisensory, or what they are, actually. I thank you for your attention, and and of course all the sponsors and the funding agencies. And I just want to announce also that we have 2 open PhD positions with deadline 9th December, 2 different profiles, one mainly EEG, intracranial EEG, and another one mainly MRI. So I thank you again. Thank you.
Thank you.
Difficult questions.
Very quick questions. Yes?
Thanks a lot for the interesting talk. One small question. Have you ever tried to use amygdala neuromeasures to predict all these fear conditioning measurements you do? So, for example, having a baseline measurement of amygdala now, let's say, and then 6 months on 1 year later, measure whether the amygdala volume, for example, could predict this.
The amount of fear conditioning of something, or the amount of response to emotion, for example. No, but I'm pretty sure that something similar has been done, and my guess is that, of course, it will, but I don't know about volume, but usually amygdala volume is correlated with direct response to emotional stimulation. So this has been directly tested, and I'm pretty sure that if it's not volume, it might be any other parameter, but for sure its integrity and will predict the amount of conditioning, fear conditioning. At least it does with salience detection.
Judith, I have a question. After you demonstrate this subcortical circuit of auditory fear response, is there like a therapeutical approach after that? Are you thinking in a translational follow-up?
Yeah, exactly. No, that would be super interesting, but I have to say that the visual subcortical route has been shown repeatedly affected in several clinical populations. For example, schizophrenia patients, or not even in schizophrenic patients, in relatives, so healthy relatives of schizophrenic patients. they, for some reason, show some sort of impairment in the visual subcortical root. Also in autistic patients, but these are the two populations where it has been mostly proven. And some have even proposed the integrity of this pathway in vision to be a potential biomarker for future, vulnerability of the disorder. But of course, I mean, I think this is super, that's something that we should, of course, consider with a lot of precaution. And, but indeed, yes, or even, because this subcortical pathway is supposed to be the one that allows you to process unconscious information, like subliminal information. And some even propose that, okay, so this would be, this would allow us to train, so you can actually train it by presenting subliminal emotional information in PTSD patients, for example, without asking them to face their feared simulation, so you can present it subliminally, but things like that. And but the thing is that both of them, so the auditory and the visual subcortical root are supposed. So this is the starting hypothesis. Both of them should be composed mainly of magnocellular neurons, and these magnocellular neurons have been shown to have very strong similarities also in neural development. And some has suggested that, in fact, if both are so similar, they might also be both vulnerable to the same attacks, the same pathogenic factors. So if the visual subcortical root is affected in schizophrenia or autism, why not the auditory one? But for now, this is just a proof of concept, and I don't know whether there is a therapeutic application.
Thank you, Judith. Thank you, and we will move on to the next speaker.
There is not so much background.
It is my pleasure again to introduce you to Gisela Sugranyes. Gisela is a psychiatrist at the hospital clinic, and she's head of the multimodal neuroimaging in high-risk and early psychosis group at IDVAPS. And she's also the coordinator at IDVAPS of the clinical and experimental neuroscience area. And we are very happy to have you here today.
Thank you very much. Thank you very much, Lupe. So thank you. First of all, I'd like to thank the organizers for inviting me to give this talk. It's a real pleasure to be close to home and surrounded by so many colleagues. Indeed, I have a clinical position. I see patients with a range of mental health conditions, but I'm also I also conduct research. And so I hope to be able to bridge kind of the gap between, well, it shouldn't really be a gap. I hope to be able to bridge the knowledge between the preclinical field and the talks we've heard so far and more of the clinical experience. So my research group is focused on trying to understand how abnormalities in brain development can give rise to severe mental conditions. Most of our work is observational. We are now beginning to delve into the experimental research with animals, with preclinical studies. But what I'm going to present today, and I wanted to say it now in case I forget later, is all observational studies. So I have no conflict of interest, and I'd like to start presenting. The title of my talk involves psychosis. So psychosis is like the paradigmatic symptom in severe mental health disorders, the two conditions which you will probably be familiar with, and we've heard a bit already about schizophrenia and bipolar disorder. Or where we're going to focus our sort of attention now. So these aren't highly prevalent conditions, but disability is disproportionately high considering the prevalence. I would like to draw your attention to the fact that WHO has stated that neuropsychiatric conditions are the first cause of disability for youth aged 10 to 24 years. This isn't just about schizophrenia and bipolar disorder, all neuropsychiatric conditions, but this, I'm giving you this figure so that you can be aware of the relevance of this as a society, of neuropsychiatric conditions in children and in youth. So why this disproportionate disability related to these severe mental health conditions? Well, the way in which we diagnose and treat these kids hasn't changed that much in the last few decades. And this is largely related to the fact that we still have a very poor understanding of the biology of the disease. This is due to many reasons, but but we think a lot has to do with the fact that research so far, at least in the field of psychiatry, has focused on patients with chronic both and adult samples, whereas we think that lots of important things which will then determine so both etiology and pathophysiology of the disease take place earlier on, coinciding with neurodevelopment. So if we go look into DSM, it's the diagnostic classification of psychiatric conditions, and look at the section titled neurodevelopmental disorders, what we'll find are conditions which have a clinical onset, a clear clinical onset during childhood. Paradigmatically, autism spectrum disorders, ADHD, intellectual disabilities, among others. Schizophrenia and bipolar disorders are in different chapters. And it is true that they most often have an onset during late adolescence and early adulthood. However, we and other researchers believe that changes in the brain and in behavior preceding clinical onset of the condition take place during neurodevelopment. So I would also like to clarify that the brain obviously doesn't follow DSM, and there is lots of heterogeneity and overlap in symptomatic presentation. Luckily, we've already heard a bit about this today. So psychotic symptoms, while we'll be studying, while we focus on schizophrenia and bipolar disorder, may also be present in autism spectrum disorders, for instance. Affective symptoms can be comorbid and can present in a range of neurodevelopmental disorders. Neuropsychological impairment is also a characteristic feature in these different conditions. And to converge finally on social cognitive dysfunction, which is what I'm going to talk about for most of the rest of the talk, is considered a transdiagnostic marker. This is kind of like the take-home message of this slide. The fact is that social cognition is impaired in adolescents with a range of mental health conditions. So even though I'm going to focus on psychosis, lots of what we are studying may be applicable to other conditions too. So I already mentioned that during childhood and adolescence, so even though psychosis typically has an onset in the second decade of life, before onset of the first episode of disease, we can individuals already display cognitive deficits, some forms of symptoms, and importantly, social dysfunction. And we and other researchers think that this maps on to abnormalities coinciding with the development of these normals of normative brain functions. So far, we've done lots of work trying to describe changes in brain structure which underpin these clinical findings, and we focused on individuals with a vulnerability for the disease, so familial high risk, and with clinical high risk for the condition. So, social cognition, as I mentioned, well, we've just seen that social dysfunction is characteristics of kids who later may go on to develop the condition. I'm going to give you an example here. stated but quite elegantly in this review by Lemmers and Johnson. So we're at a party. A friend just finished a phone call, and he's had bad news about a recent job interview. He hasn't shared the news with us. He didn't want to tell us about the negative outcome. But he comes back, and we see this facial expression. We could discuss whether he's sad, upset, but my interpretation is that he is angry. So this would tap in to one of the first social cognitive functions, which may be abnormal in these conditions, which would have to do with misperception of an emotion. I don't think there's a reason to be angry. It's unreasonable. So I'm not understanding social context. He should have fun. We hardly ever see each other. This taps into theory of mind. I'm not understanding his perspective. I'm not making a correct inference of his mental state. And this will lead me to elaborate a wrong conclusion about his mental state and his behavior. I think he doesn't enjoy being out with me. And this would lead to a wrong attribution, jumping to conclusions which could be at the basis of suspiciousness, resentment. And the consequence of this is stressful, I better stay in by myself, I won't come to the party next time. So possible consequences of social cognitive dysfunction may lead to social anxiety, stress, defensive behavior, and ultimately social isolation. So, as I just illustrated, social cognition is composed by, can be categorized into different dimensions. The one which may have been most studied, apart from facial emotion recognition, which we've heard a bit about already, is theory of mind, and it seems to be the one which is most correlated with community functioning, so with social everyday functioning in people with non-affective psychosis. So this is, quite a few studies have been performed, especially in adult chronic schizophrenia patients, and especially using a task-based approach. So in the scanner, the patient is has asked to conduct a task, looking at reading facial emotions, trying to infer mental states with their theory of mind task. And this is a summary from a recent review looking at the different brain areas which are activated during these tasks usually, and which show deficits in patients with schizophrenia. So specifically, just to summarize, emotion processing, amygdala, sensory cortices, theory of mind, medial prefrontal cortex, and temporoparietal junction. But what we saw at this point in time was that we knew very little about what happened when psychosis had an onset, when these functions, when social cognition was actually still maturing. And that is what happens in cases with early onset psychosis, so psychosis with an onset during adolescence, or clinical high risk for the condition. So I did part of my thesis on this in London a long time ago. And when I came to Barcelona and I started working with patients with psychosis, I asked my supervisors here, Professor Castro Fornieles and Professor Valleza, who are psychiatrists in the hospital, if we could perform similar tasks in the scanner with our patients with psychosis. And they said, no way, you're not doing that. We will lose all our sample. We work with cohorts of youth with risk or with first episode psychosis, and they said there's no way they're going to be able to sustain this kind of task. But I still wanted, I still thought it was really important not just to look at brain structure, but also to try and understand the brain synchrony behind these elaborate cognitive tasks, which explains so much of social function. So I did convince them to incorporate a resting state scan, which we still use today, and which I will focus on over the next few slides. So research in this kind of population has been ongoing for over 2 decades now. We have, and both locally and also in collaboration with several international consortia. We have kids with parents diagnosed with bipolar disorder, schizophrenia, kids with clinical high risk for the condition, or with the first episode of psychosis. And we assess them over time with clinical tools, so questionnaires, tasks, MRI scans, and blood. So first of all, there was our first study. What we set out to try and understand was how resting state networks developed over time. There was very little knowledge at the time on what happened in adolescent brain development in healthy adolescents. And this study was conducted by Cristina Soler Padulles, who's currently here at the university, and who was in our team quite a few years ago. And what we found was pretty robust age effects on the default mode network. So default mode network is the network which activates, deactivates during tasks, and tends to activate during rest, and which is one of the most studied and replicated networks in resting state designs. And we found these pretty robust age effects, especially in the anterior default mode network in the medial prefrontal cortex. And we also found age effects in the language network too, and surprisingly few sex effects, which we were expecting to find. And this was in 130 healthy controls. We pooled samples from different groups in the child psychiatry department of the hospital here. Then Cristina conducted a case control comparison. She selected the language network because at that time lots of evidence supported the idea that we needed to go to the frontal and temporal lobes to try and tease apart illness-related effects and psychosis. She compared 34 patients with first episode psychosis, always with an onset before age 18, 44 clinical high risk, and 35 controls, and found and identified within this language network a cluster of reduced connectivity, intrinsic connectivity in the right inferior frontal gyrus. Interestingly, this region of reduction in connectivity was associated with a measure of white matter integrity measured with tractography in the same region. So pointing to an association between functional and white matter, so functional and structural connectivity in this region. So we then incorporated in the team Dani Izarbe, who's here in the audience, and he conducted his thesis sort of delving into a deeper level in the sample and trying to compare the resting state data with theory of mind tasks. So as I mentioned before, theory of mind's capacity of understanding that others present their own beliefs, intentions, or desires, and attributing mental states to others. This is a graph which he presented for his PhD, which is sort of pooling the evidence from different healthy controlled studies over age. And as you can see, theory of mind performance increases over time and especially during adolescence. There's evidence suggesting that connectivity within the default mode network is associated with theory of mind performance, specifically the temporoparietal junction and medial prefrontal cortex. This is evidence from adult samples comparing out-of-scan behavioral performance with connectivity of the default mode network. So his first study was to compare 27 individuals with the first episode of psychosis and with 41 controls. First of all, they conducted this theory of mind task outside of the scanner, the reading the mind in the eyes test, where an individual is presented with two eyes and they have to infer a mental state. So it's not about reading emotions, it's about inferring the mental state behind these eyes. And it's compared with a control task where the individual has to inform on the gender of the eyes, the sex. And after performing this task, they are invited to the scanner where we still use a sequence on the 3 Tesla scanner here in the labs, where we acquire 8 minutes of resting state fMRI. And we use a template in order to identify the default mode network. We also did a secondary analysis separating patients with psychosis according to whether they develop schizophrenia or affective psychosis, which includes bipolar disorder, over time. And what we found was that in a group by condition model, so in essence, we found a group effect in patients with early onset psychosis who perform more poorly in this theory of mind condition. And we also found a significant interaction between performance and age, whereby controls increased their performance over time, as we'd already seen from the literature, whereas patients with early onset psychosis didn't have a developmental gain in the ability in the theory of mind task. When looking at the MRI results, we found that within the default mode network, there was a cluster in the medial prefrontal cortex showing in which early onset psychosis patients had reduced connectivity in comparison with controls. In a very close by cluster, we found a group by age interaction, whereby controls, as we'd already seen in the literature, increased connectivity over time, over age in the medial prefrontal cortex, whereas patients with early onset psychosis decreased connectivity in this region over time. This is a one data point per subject, so this is cross-sectional data. It's important to bear this in mind. This isn't changed. This is group by age effects. We also wanted to try and see how much the functional connectivity assessed in the scanner could explain the relationship between group and theory of mind through a mediation analysis. And we found that approximately 17% of the variance or of the effect of group on theory of mind was explained by the signal which we detected in the scanner. This is obviously statistics. Okay, but it was important for us to understand that the functional connectivity could be explaining a portion of the group effects in behavior. When subdividing the sample according to diagnosis, we found that only individuals with early onset schizophrenia showed reduction in theory of mind performance in this behavioral task, and not patients with early onset affective psychosis. Whereas in the scanner, we found that the group's effect in functional connectivity in the medial prefrontal cortex were present in both diagnostic subgroups. And just to say that these results held when we controlled for general IQ, socioeconomic status, medication. So the next question was, so what happened? When do these deficits arise? And what about kids who who are at risk for the condition? Do they already have these behavioral deficit and changes in MRI scan? We had this a sample of 50 individuals with clinical high risk for the disease. So we consider them at risk because they have attenuated psychotic symptoms. So not to the clinical degree of psychosis. They don't need to be admitted into a ward normally. They don't need high doses of antipsychotics, but they do show subtle changes in perception, in behavior, and thought processing. And we consider them, and approximately 20% of these develop a psychotic disorder in the next 2 years. So we followed them up for 2 years, and 15 of these kids developed psychotic disorder. This is a 3-group design where we divide the clinical high-risk kids according to whether they develop disease or not, in comparison with controls. And we did exactly the same protocol with the behavior and the MRI scanner. And in this case, we didn't find group effects. We didn't find that the groups were essentially different. What we did find, though, was a group by age interaction, whereby healthy controls experienced this increase, this developmental gain in behavioral, in theory of mind functioning, whereas in clinical high-risk kids who transition to psychosis, it didn't improve their performance over time. It was as if they had a developmental arrest in theory of mind capacity. And we saw I'm nearing the end, and we saw very similar effects in the brain imaging. So we found a group effect in the medial prefrontal in a very similar cluster to the ones we found in psychosis. So clinical high risk for psychosis. Individuals who transitioned to the disorder showed reduction in intrinsic connectivity in the medial prefrontal cortex. And this wasn't present in those who didn't develop the disease, okay? And we also saw an age by group effect here, whereby controls experienced an increase in medial prefrontal cortex connectivity, which wasn't present in clinical high-risk patients who transitioned to psychosis. We also conducted a survival analysis and found that, and by dichotomizing medial prefrontal cortex connectivity, we found that showing low functional connectivity increased by six the risk of developing a psychotic disorder over time. So I'm getting into my sum up slides. So patients with first episode psychosis, specifically those with schizophrenia, showed poorer performance in theory of mind than tasks and controls. And both patients with first episode and high risk for psychosis who transitioned to the disease seemed to show this developmental arrest or delay in the acquisition of theory of mind skills. So Danny, in his thesis, also drew the line for ASD. This is autism spectrum disorders, which would, some authors suggest that in adulthood show similar deficits to patients with schizophrenia, but the timeline of differences is so the clinical course of the condition is different. In terms of brain functional connectivity, we found a very similar pattern between psychosis and clinical high risk, suggesting that the disruption in medial prefrontal cortex connectivity in the prefrontal, in the default mode network is already present before clinical onset of the disease. And this is kind of like the big take-home message here. But in the clinical high risk, this is still not impacting the theory of mind performance. So it's probably a disruption which is starting locally and progressing as psychosis develops. And this is something that we need to explore. We found these interesting diagnostic subtype differences between early onset schizophrenia and early onset affective disorders, which we have explored in a different study looking at dynamic resting state fMRI conducted by a postdoc in our lab, Mireia Macias, and which in 2 words, basically what she found is that brains in early onset schizophrenia patients are less efficient and more immobile in comparison to patients with affective psychosis, but I won't go into that now. So, implications. Brain functional changes occur prior to impact on behavior. There may be a transition from more focal to more global changes as illness progresses and also according to illness subtype. And I think the clinical implication here would be the possibility to stratify youth according to their brain functional phenotype in whom to target treatment efforts, so social skills training. So to use the information from the brain MRI scan to inform treatment selection. So future research taps into limitations of our current design. As I mentioned, we have 1 data point per case. We need to see if this holds when we look at repeated measures, so longitudinally, and going even further back in age, looking at even younger samples. And this is ongoing work by people in the team. We also obviously need to validate these findings. We need to know to replicate them in larger samples, and we are participating in the ENIGMA initiative for testing the brain functional connectome in clinical high risk for psychosis patients, which we're super excited about, and also validate with just try and understand what all this means at a smaller scale. So we are currently conducting molecular imaging in these same participants and using both human and animal models of receptorial dysfunction and trying to understand the relationship with these findings. And that's all. Thank you very much for your attention. We are also hiring, so please contact me for postdoc. Thank you.
Thank you, Gisela. Impressive amount of work. It looks easy, but this is a lot of work, and these are not mice or rats. These are human beings, so that's a lot. Short question for Gisela. No, I was wondering, Gisela, if you find any sex effect on your results.
We looked, because this is a really important question. In fact, the first study looking at development in healthy youth, we tested sex and age by sex interaction, and we saw very little sex effects. We thought that maybe it's one of the potential reasons is that maybe we like a critical period for understanding sex effects is puberty. And our sample is a little, we definitely do have participants pre and post puberty, but it's the largest, later on is more highly represented in our sample. So we may have lacked power to see the difference between all the sex effects and the pre-post puberty. I think it's a fascinating question. And Danny was super rigorous, and he looked at sex effects in all of his models, and he didn't see an effect there. We can't rule out that this isn't a false negative finding, in that the sample sizes are modest, so I can't assert that there's no sex effects, but we didn't power the study for this. We looked at it as a confounder.
Thank you. If there are no more questions, we will move to the last speaker. Àngels Permuy, thank you, Gisela. And before introducing Àngels, I have to say that Àngels will give the talk in Spanish. If there is anyone that doesn't understand Spanish, we have, raise your hands, because we have some translator machines. So you can follow the talk. So there's a raised hands there. Raised without. There are more there. Don't be shy. Raise your hand. Cristina, on the right. I will do the introduction in English, and then I will give her the word. Àngels Permuy is a special education teacher with a degree from the University of Barcelona, and she has also a master's degree in applied behavior analysis. She is teacher at the Fàsia Sarrià school here in Barcelona. And she will talk us about how, like, we are focusing out in this session, and now she will be able to tell us, like, in the practical side, how to deal with these special needs children.
Thank you very much. Angels, you have the floor. Good evening. Thank you for being here. For me, it is a big pleasure. I can participate here in this event. I'm not a scientist, but I do hope to give an important talk. I want to talk about the impact of neuroscience. I am a teacher. I have been working for 20 years in this school. We have 75 pupils, and 70% have a diagnosis of autism. The prevalence in autism has been increasing, and the prevalence is of 1 out of 100 children. In Europe, the prevalence was 1 out of 57, and in the States, it's 1 out of 36. So these are important data for schools. Those people need support, and society should be ready to assist them in the better way. If you didn't have the opportunity to know autistic children, I would like to give a little bit the context. In 2013, we have the DSM-5, and for the first time, we had under an umbrella different diagnoses. They had similar symptoms, but they were Asperger, for instance, you know, autism, TGV, etc. And for the first time, so there is the spectrum of autism. And there was the necessity of dividing all these populations. So level 1 was the least severe, and level 3, but 40% will be in level 1, and the rest will be between level 2 and level 3. So this is a very high percentage, and they will need a certain support in order to learn in schools. What does it mean to have autism? So here, people have specificities in 2 areas. One of them is communication and social interaction. We can find children who have difficulty in order to understand verbal communication, and sometimes they cannot understand behaviors in other people. Of course, those are difficulties in order to start communication at large. So it won't be the same. So if someone belongs to level 1, it's not the same. In level 3, there are children who don't develop language, and they need added methods. So this is a handicap for the children, for families, and for people. And then we have behavior patterns. So this is very worrying with families. So there are children who have difficulties in order to stop an activity and to do something else. Children that can have a stereotype. For instance, they move their hands, and all those behaviors can even become a barrier to learning. And we will find as well people who have important specificities in the sensorial area, hyper sensitivity, and hyper reactivity. For instance, there is something that impacts me. So this is feeding. Some children have stopped eating because they cannot tolerate a certain temperature, and it becomes very difficult. So sometimes when they change clothes, or when they wear sandals, or when they wear another type of shoes, this can become a trauma for the families and for myself. So those children need a special support. So what is the impact of neuroscience? Neuroscience is giving us a way, a path. And thanks to research, we have isolated genes that are implied in autism. We know that they are genetic variables, syndromes like the reality and epigenetics. Sometimes the environment can play a role in the development of autism. All this helps us so we can have a support for diagnosis. Not very long ago, the diagnosis was only the observation of a psychologist. The child was observed in its deficits, and with an interview, the diagnosis was established. So now we have genetic variables. This is important. Not very long ago, there were theories, and in those theories, it's like fridge mothers, or people thought that vaccines could cause autism. And now, of course, we support diagnosis, and we are more sure. We know that the diagnosis is not wrong. And with neuroimages, we have found alterations in the brain connectivity. So we know that there are areas that are more or less developed. And at the end of the day, the way they have to understand the environment goes one way or another. There is an imbalance in the neurotransmitters. So if science can advance and can research all those domains that are open, I think that we could improve the quality of life of autists. This is what is worrying for us. All the research for therapy create a place for the therapist. If you understand that a child generates a behavior, it doesn't mean he's difficult, because we will have another vision of the child, and all this will change. So how do we go from diagnosis to intervention? So with neuroscience, we learn that neuroplasticity is very important. So it is more effective in an early age. And more than that, we saw on the other slide, we can understand that there are not two identical autists. So the intervention should be individualized, customized. What are the therapies that we have? We have a lot of them. This is as many as psychologists. So those are the ones with more scientific evidence. What do they have in common? They are intensive. So they comprise from 15 to 20 hours a week. They are recommended for very small children. So they imply communication and games, playing. So it means that they should have more social will of communicating, and families should participate in all this development. Otherwise, therapies lose value, and therapists should disappear. So families have to know how to manage the behaviors of the child. and they have to guide them. There are other promising therapies, they are emerging. So we have interventions based on technology or occupational therapy based on sensorial integration. So we have to base them. So neurofeedback is used in autistic persons. What is the situation in Spain? Spain, and namely in Catalonia, where I live. If we compare the current situation in Spain, and of course we lag behind. Diagnosis comes very late. Of course, we can have an early diagnosis when the severity of the illness is stronger. So there are pediatricians who don't know the symptoms of autism. They don't know it very well. So we have pediatricians who ask the families to be patient because they think that the development of the child is going to come later, and we are losing here value. And sometimes there is this early intervention. So all this, all this intervention is private. They are very, very expensive. And in the public services, families are given 45 minutes in an office. And the interventions should be done at schools, at home, in a park, in a supermarket. And the early intervention here in our country is an office, and very often the family is not included, because if the mother comes in, the child is distracted. So it is difficult afterwards to apply interventions. And autistic children go to school, but they are not well prepared, and we do have a huge problem here. So what are the challenges for the families? They have to fight in order to have a diagnosis. They have to manage emotional health. They have to find access to services and resources. Sometimes the families are exhausted. They have to go to the school so that the materials are adapted. And sometimes they need training, ongoing training. The system doesn't support them. So what are my conclusions? First, we do need neuroscience, and we have to advance. We have to understand the functioning of autistic brain. Therapies should be individual. Unfortunately, many interventions are general, and sometimes what happens is that the children don't learn. We don't have to forget that all this is to improve the quality of life of people and of the families. So without research, we cannot do it. We do need those interventions and advances in research. Thank you very much.
Gracias, Ángel. No tan rápido. No sé si hay alguna pregunta para este broche de oro en la sesión.
Hola, Ángel. Muchas gracias por la charla, en primer lugar.
Thank you very much for your talk, Angel. I think it was important because sometimes I have worked on autism, but sometimes with animal models, and you do forget that there are people who are living this condition. I would like to ask two things. At the beginning, based on your experience, so there is this prevalence is increasing, and in your perspective as a teacher, do you think it is because we work a lot for diagnose, and perhaps those children were not diagnosed. People thought that this child was difficult, and now these children are in a special school. And then you see that you have described 3 levels. Are there more children born autistic? I can be wrong, but honestly, I think that they are more and more. It is true that the umbrella is bigger, but when you know an autistic person, you realize that the brain functions differently. And children in level 1 were not under this umbrella. And now, of course, this gives problems. This gives rise to problems of anxiety, and the diagnosis should be less. And now I am a counselor in ordinary schools, and you can find them in every classroom. I have a 13-year-old daughter, and they were not. And now in the smallest classes, they have 2 or 3 in every classroom. So I do think that this is increasing. I don't know if this is due to food or to something we do, but I would like you to do more research. And yes, we have to talk as well of prevention, because sometimes the children can go to one level to the other if the skills are more preserved. This is a reality, but it would be very good to work on prevention so that the level of severity is less. So this would translate into quality of life. And here, I don't know if you know it. You know that I know that you have a lot of work in schools, but you talked about therapies. Or do you know other therapies? I don't know. So here, those are children, but these children will become autistic adults. Is there any therapy self-managed? Of course, so that themselves they learn to manage with the comorbidity. Of course, it will depend on the level of severity. In level 1, when they are children, all the children need care and guidance for hygiene, for preparing things to go to school, etc. So the parents are very important, whatever the level is. In adult life, I do think that a person in level 1 can self-manage the therapy, but in level 3 of severity, this person will be dependent, and of course he will need help with therapies. We say that families play an important role, but yes, they play an important role in all the children of the world. For me, to separate family from therapy, from the child's education, for me, it is very difficult. Thank you. Thank you very much. I loved your talk. My Spanish is not perfect. I also work with animal models, and sometimes we forget what we can do. Thank you. Thank you very much for reminding this. And I would like to ask a question. When you teach, do you see if there is a difference between girls and boys? There are many hypotheses. They say that there are more boys than girls, but there are people who say that girls are more able to mask this. So there is, I'm not a scientist, so this is what I read, what I can see. Up to now, yes. I wanted to talk about this in the presentation. So here we say that there was one girl and 4 boys. I have a classroom with 9 students, and there is only 1 girl. And they say that women preserve all this social part better. We are more social than men. And in the development, when you can see, so those children are 3 years old, and you can see the difference in language. Of course, there are exceptions, and there is a boy who learns to speak very well and very rapidly, but the girls develop language earlier. So there are more theories than autistic women are more difficult to diagnose. Even it was not my intention if we want to be feminist. So the batteries of tests are made by men. So of course here we can open a wide debate. Yes, perhaps, yes, it is possible for women to mask this condition. Thank you very much for your talk. It was very interesting, and thank you because we have here another perspective. You mentioned interventions that are very intensive with many hours a week for years, or of course those are early interventions. So this is from 2 to 5 years, and one of the therapy which is PAD, which is until 10 years. So, and then it is in order to go to a more complex communication. But all this happens when they are children. Could you give us some examples? What kind of improvement can we expect? Of course, it depends on the case. Yeah, but basically, autism entails difficulties, of course, the management of motivation. There are children that have restricted interest, and sometimes with more severity, they don't pay attention to the environment. So those therapies are based on the management of motivation, and they use games in order to open a field towards to other discriminative of the environment. For course, the interventions have to be early, and is it possible? Is it possible to obtain improvements? Of course, there is a genetic part that exists, and a part of cognitive skills. In some countries, people sustain that there is an improvement of 15 to 20 in IQ. So this is very important. I think that, yes, we are late. I didn't manage time, but thank you very much. Thank you very much for everything that you explained here, and we did feel very useful here. Thank you. Thank you very much for your attention.